Do you know? It looks like the presentation button is not working. So this one? Is that the presentation? Does it say present? Would you mind helping out?
Is this PowerPoint?
No, it's something else.
It's Google Slides. Yeah, you can go ahead. Oh, okay. Thank you. Hello, everyone. So I'm going to talk about DEEPCON, a method for protein contact prediction. So for those of you who do not clearly know what the protein contact prediction problem is, the problem is given a sequence of amino acids, that is a protein sequence, we would like to predict how close each pair of amino acid is with each other. So ideally, this would be a distance map, but then if you set out a threshold on this distance map, and all the distances below a threshold are 1, and everything above that is 0, then you get a contact map. So the problem is, given this amino acid sequence, we would like to predict the contact map. So the problem is very similar to a computer vision problem, or even similar other problems known as depth estimation, specifically something like a monocular depth estimation, or even image segmentation. So in computer vision, the depth estimation problem is given an image like this, which has 3 channels, red, green, blue, or RGB, HSV, we would like to predict the depth intensity for every pixel in the image, right? So, for example, you can see that the sky is very far away, so that's why the color is red, and so on. So the contact prediction problem can be formulated, in a way, formulated to be like a depth estimation problem, or even the image segmentation problem when we're doing the contact prediction. So from a machine learning perspective, there are some features of this problem that make it very unique. The first is, for protein contact prediction, the number of input channels is really large, unlike images where we usually have only 3 channels as input. And this is not something new. Other places where, for example, taking plants images, where people take hyperspectral images, we observe this lot of input channels as input features. Another unique feature about the contact prediction problem is, unlike images where we always have 2D input channels, in case of proteins, we may either have sequence features, 1D features, or scalar features, and 2D features. Unlike problems in computer vision, even though, in a framework setting, it's like a computer vision problem, visualization is extremely difficult. Unlike images where we can visualize even what filters are learning and use them eventually to improve the overall prediction, visualization is extremely difficult. There is one group who has done a little bit of work on that, but overall, visualization is difficult for contact prediction. That means that it's really difficult to dig into your deep learning model and investigate what the models are learning. Another unique feature of the contact prediction problem is the non-scalability. For example, if you have an image database, you can scale one of the images in the image database, and still that image becomes a legitimate, genuine image. Whereas in case of a protein database, you can't pick one protein out of the database and scale it and make it bigger and still have it going. That's because the units of the protein structures, no matter how big or small the protein is, are always, from a distance perspective, of a fixed size. Also, unlike images where we always may have a fixed size input, or we can scale the input to a fixed size, proteins have variable size input. For example, some proteins can be even 1,000 residues long, or even longer than that. Unlike many image segmentation problems, we have limited and unbalanced data. That is, the positive classes in the output contacts is really low. And yet, unlike image segmentation, where you would like to segment each object clearly, we don't need all of the top predictions, all of the positive classes that we predict. Also, we have to bear in mind that the overall goal in the field is to predict continuous distance values, and contact prediction is simply a way to simplify the problem so that we can attack it. Also, long-range contacts are more important, so the contacts in the upper triangle in the contact map are more important, and also the overall contact matrix, the output matrix is symmetrical. So these are some of the features from a machine learning perspective which makes this problem different from depth prediction or image segmentation problem. So why predict contacts? CASP, if you look at CASP 13's website, they put out links to all these really nice articles. The bottom line is, so far, although there are many other promising methods, so far, contact or distance prediction holds a key to solve the overall protein folding problem. So now let's look at the overall flow of a contact prediction pipeline and see where we can attack to improve it. So the generic pipeline for a contact prediction takes a sequence as input. We predict multiple sequence alignment, right? And then we also predict 1D and 2D features, for example, sequence profiles, secondary structures, either using the multiple sequence alignment or directed from the sequence. And then we translate, transform, or calculate feature engineer to prepare the data in a way that's ready to be fed to a machine learning pipeline. And then we train a deep learning model, predict contacts or even distance maps, and then use tools like Rosetta, Confold, or even the talk that we just saw before to build 3D models. So for deep learning, learning to predict contacts, we have the truth structure. We calculate the ground truth from the truth structure, and then using loss function, train the model to accurately learn to predict contacts. So in this pipeline, let's look at the places where we can attack to improve the overall contact prediction performance. So the first place we can attack is definitely increasing the quality of multiple sequence alignments. The more deeper homologous sequences we can find to generate a better multiple sequence alignment, the better contacts we can predict. The second place we can attack is developing a right deep learning architecture, an architecture that's customized and that's best suited for the contact prediction problem. The next place we can attack is feature engineering. So in yesterday's talk, we saw that some groups discussed about encoding. So that's one way we can do some feature engineering. How do you convert these sequence features to 2D? Whether we should calculate covariance or precision metrics. So all of these techniques where we prepare the input data in a way such that the deep learning model finds it easy to learn would be another place to attack. The last place I feel like is a good place to attack is using the appropriate loss function. Some of the unique features that I discussed about the protein contact prediction problem make it very different from other binary classification problems or even other regression problems like depth prediction. So going besides binary cross entropy or MSE should be a place where we can significantly improve the performance, because out of all the predictions we make, we don't use all of them. We just use top L by 2 contacts. So what's interesting is of all these four places that I showed, 3 of them are basically places where deep learning can bring the progress. So I'm going to focus on one idea to improve this area where we can improve the existing deep learning architectures. So in the most recent CASP, what we saw was the top performing groups, AlphaFold from DeepMind, Dr. Jones' group, Dr. Su's group, and Dr. Zhang's group had one thing in common, and that was all these methods use residual networks to predict contacts or distances. So the question becomes, are residual networks the final architectures that are best fit for this problem, or can we improve them? So in this project, we investigated if we can improve residual network architectures to further improve contact prediction. Here's a typical residual block that has 2 convolutional layers, batch norm layers, and ReLU edge activations. Of the many things we tried, we also tested replacing the second batch norm layer with a dropout layer. We also tested, as DeepMind tried, replacing the convolutional layer in the second convolutional layer with dilated convolutions with alternating dilation rates of 1, 2, and 4. And we tried to combine these to have a dropout and convolution with dilation. So then there are two kinds of features within in the field of contact prediction that we recently see we've been using. One I call the sequence features, where you predict things like secondary structures, sovereign accessibility, sequence profiles from the sequence, and then obtain various channels to be fed to the deep learning model. The next category of features is using the multiple sequence alignment as input and directly calculating features such as covariance and precision metrics. So if an architecture works well for one of these input features, it should work well for the other, right? So to find an architecture that robustly works well, it has to be tested in either of these two input features. So we tested both of these. We created two setups where we use the DNCon2 data set with around 1,400 proteins, and we generated sequence features, and we use the DeepCov data set, and we use covariance features as input to evaluate various architectures. So when we use covariance features as input, what we find is if we evaluate top L by 5 long-range contacts, a standard residual network and a residual network with dropout perform similar. However, if we dig deeper and look into top L long-range contacts or even all medium and long-range contacts, we find that adding dropout to a standard residual network actually significantly improves the performance. What's also very interesting to note is the unit architecture is very common in computer vision, and it has been widely used. What we find is, for this particular problem, probably because of many of the features that I discussed earlier, unit architecture performs even worse than a fully convolutional network architecture that has much lesser number of parameters. Same thing with sequence features as input. So a residual network is better than a fully convolutional neural network, and residual networks with dilation perform better than residual networks, and residual networks with dropout perform slightly even better than residual networks with dilation, and then combining dilation and dropout actually significantly improves the performance. Once again, this is only in top L by 2, so if we look into more contacts, that is evaluate top L or even all medium range or long range contacts, this improvement is much more amplified. So I'm not here to say that DeepCon is the #1 contact predictor. But the lesson to learn is that there is so much improvement that we can make just in the deep learning architecture. So here's a table that compares DeepCon with other methods, say, for example, DeepCove and PCon-C4, trained exactly or in a similar data set, but use a different deep learning architecture. You can see that just by changing the architecture, we can gain so much improvement in performance. And we have CCM paired and free contact as a reference. I also tried feeding the alignment. So these are alignments from a much older method, DNCon2 method. And I also tried feeding the alignments to the RaptorX web server, which allows you to upload alignments as well. And with the exact same alignments, RaptorX obviously performs better than all other deep learning architectures. And I believe the reason why RaptorX performs better is because, of course, it uses ensembled models, it has larger training data set, and it also uses many other features like sequence profiles and secondary structures. But the focus here is to compare methods that use single deep learning model, and what we see is when we have a single model and we use a fixed training data set, there is still a lot of room for improvement in the deep learning architecture. So overall, is there any room for improvement? So if I see the progress in protein structure prediction as a river flowing, right, then the banks that support the progress will be definitely deep learning and correctly exploiting the sequence data. So both have to go hand in hand in order to push the progress. So here's an example. So if you simply Google or search dropouts and convolutional neural networks, here are some of the articles that you will run into. So basically what you will find is dropouts do not work well with convolutional neural networks, right? But what we just saw is, at least even if it is slightly higher, it is an example where we could improve existing methods that we've been using. In fact, from our keynote speaker's talk to this talk, just in half an hour or so, we saw a percentage or two improvement just by adding dropout to the residual architecture. So in conclusion, what we see is residual networks are not the final architectures for predicting protein contacts. The results in this work are an example. At least something like NashNet that people have done for, Google has done for computer vision, at least must be tried. Methods such as capsule networks could hold a lot of promise for solving this problem. And counterintuitively, architectures that work well for image datasets may not work well for contact prediction. And unit architecture is an example where the unit architecture has around 35 or 36 million parameters, and it performs worse than a fully connected network that has less than 1 million parameters. And the way I see it is contact prediction problem is now a problem for both domains, that is machine learning experts and bioinformaticians. So with that in mind, I've created a challenge, protein distance prediction challenge, and it's under the IEEE ICMLA 2019 conference. There is still 2 months deadline to participate in the challenge. What I've done is put out everything that you need, including the data set, how to evaluate your predicted distances, and input features and everything, and a very tiny deep learning model as well. The challenge is for machine learning experts to not worry about the domain knowledge and work on the machine learning pipeline to see if you can make as much improvement as possible. I'd like to thank people who've supported in my research, and I'll be happy to answer questions.
This is a comment not just to you, but to all the speakers this morning and all the other people interested in this problem. And I wonder if there is something to gain from not modeling a single structure from the family sequence alignment, but actually modeling all the structures, because all of those are real proteins, and maybe there is some information to gain to improve the prediction of any single structure from predicting all of them, not just one from the sequence alignment. Is that something that makes sense at all, or?
I didn't quite get your question.
You can answer, but anyone else has probably a good answer as well.
So I think something important to remember here is that almost all the input features are derived from the alignment, which would be the same across the family. And so actually what you find is that the structure you get out would be almost the same across the family. Using.
But all these sequences, all these individual proteins have differences that affect whatever potential you're using to minimize the
Yeah.
structure.
And that, I'd say, is one of the main limitations of this method, because you can't really get to that level of granularity if you're using alignment-derived features, unless you do a kind of refinement at the end, which is based on the actual sequence.
Just a small comment to the same thing. My guess is that actually the time-consuming part is often the folding. I mean, generating the MSA can take some time. The deep networks takes minutes or seconds, and then folding is what takes maybe an hour or something like that. So that's really, to generate many structures would make it quite much slower.
Hi. I have also a question sort of to all the speakers. Have anyone ever tried to look into which features are most predictive, most contributing to the performance of the models, sort of interpret the deep models, and maybe we can gain some biological insights from that?
So to answer the question, of course, once again, when we build a deep learning model to make a prediction, right, we're still yet to really investigate and fully understand what exactly a deep learning model is learning, right? That I'd say is an active area of research. I think all of us are interested to really know that. There has been one work that I know where people have tried to understand the first layer of what the first layer of convolutional filters learn, but otherwise I think it remains largely ununderstood what and how these models are learning biologically to make these predictions here.
Thanks. Brief question about the batch normalization. You didn't mention how the batch normalization affects the final results. Yeah,
I think I didn't even test it actually. Yeah, Because
the batch normalization somehow serves as a dropout. It's a kind of regularization too.
Yeah, so they're both, if we don't have batch normalization, of course, we can't go very deep, even when we have residual networks, because there are problems that come in. But yeah, combining, as you said, combining the two regularizations actually, that's what I show, it improves the performance, yeah.
Thanks.
Okay, thanks again. All right. In my mind, we only can do this sort of stuff if we really find out whether methods work or not. And of course, I'm a big fan of CASP, but I have to say that Cameo really, in a quiet way, is doing a lot more testing of how well methods work than CASP is. And so it's great to have Jürgen Hess tell us about that.
Thanks, John, for the nice introduction. And welcome.